I'll be giving a talk about structural and transcriptomic comparisons between soybean reference genomes and soybean Australia wild relatives. And during the course of my talk, I will really be talking about a lot of different glycine sequencing projects. So I will have this nice picture figure in between each one so you can tell when I'm switching from different projects. The first one I'll talk about is, of course, about the Australian relatives. And this was named SoyMap II, Leveraging Genetic Diversity. And this worked with 6 different wild species from Australia. And over the course of the project, we developed a draft genome for syndetika, and then 6 genome-guided transcriptome assemblies. This is basically a really big collaboration. There are a number of people working on this. Of course, my supervisor, Stephen Cannon, the PI was Scott Jackson from Georgia. And these Australian relatives are very unique. They're very different. And they come from many places throughout Australia. And you can see just basically like how the pod structure is different, flower, and even like the unique leaf structure and how they're very viney. And of course, we have worked with several more. Syndetika, again, very different structure. And then tomatilla, you can see, has this really cool kind of bunched up flower structure. So it's a unique thing to work with. And you can see how this is actually spread in Australia, even to Papua New Guinea. And this is a location. You can use this nice map on our website, and you can locate. These are all the ones that were collected throughout the project. And here's all 6. And they throughout basically cover a good portion of Australia. So through the sequencing, what we did is we used Lumina sequencing here at Iowa State University. About each species, we got about 230 million sequence reads, paired end. And we decided to use the Trinity genome-guided transcriptome assembly. And we used the Glycine Max version 2 assembly and also the syndetika draft genome assembly as the genomes to guide these transcriptomes. And then, of course, we had to filter this because if we know through some RNA-seq research, sometimes you do get some chimeric sequence reads, so you need to filter this out. And you can see in the table, here's a number of unigenes for each one, and they're characteristically about all around a little bit over 60,000. But then syndetika right here, which was mapped, basically used with the syndetika draft genome, and it is a draft genome, does have fewer, but it's a draft genome. So we're still looking into seeing if we can get this back up, see if there's genes missing in the draft genome within the Glycine Max. What we did in the first thing is look evolutionary, really, how do these relate with Glycine Max, to look at the evolutionary differences, because that's one thing that's really interesting, like how is this really different between the Asian variety in Japan, Korea, China, compared to Australia. And our graduate student Jacob did this work. Whoop. And these stars right here basically just indicate the split due to the whole genome duplication. And what we found is that the Asian variety, Glycine Max, did diverge from the Asian variety, or the Australian variety, about 5 million years ago. That's an estimate. Of course, this is after the whole genome duplication. What Jacob also found was that within the Australian varieties, there is some species that are diverging faster, like evolutionary diverging faster. The ones that are in squares, the tomatilla here, is actually diverging faster than the Dochacarpa. The same here, where the species is diverging faster than this, and it's for both. And then you see it's for both whole split between the two. So it's kind of a nice way to see that there's some evolutionary distance between these two. So now about the draft genome, and I wanted to emphasize this is a draft genome, and it's one of those things where I find it interesting, and there's so much to find out. But it's, as you can see the numbers, it's a draft. So the total number of scaffolds is a little bit over 14,000. And then if you remove all the ones that are less than 10 kb, you only get a little bit less than 7,000. So about half of them are too small to work with. So I have to throw half of those away. And then to make things more complicated, I have no linkage map. I really cannot make a reference genome out of this. So we basically just made a synteny map. That's all we really can do. And some of them did not map. About a little bit over 1,500 didn't map. And then some were removed because for quality control to make a nice pseudo molecule. The one thing we decided to do with this draft genome was to really look at the structural rearrangements. We know that soybean genome has many different types of chunks in it because of the multiple whole genome duplications. So can we look at this and answer some evolution questions? And you've seen this figure many times in different types of publications. And then if you go to Soybase, you can find the synteny viewer basically here on the gbrowse. So what's the question? When did these structural rearrangements occur? So we estimate that the whole genome duplication happened about 10 million years ago, and then about 8 million years ago, polyploidy. These are all estimates, there are debates, but let's just go for today. But then when did these structural rearrangements occur? Did they occur early, when there'd be shared breaks within both genomes, or did they occur late, after Glycine max and Glycine syndetika speciated out? So we basically looked at, are the homologous junctions between Glycine max and syndetika the same? So we look first at Glycine max. We can see that there are 208 junctions, basically these breaks. We're looking at these breaks right here. As you can see here, it switched from a synteny of 16 to 9. So we're basically counting these breaks and looking at them. And there's 208. We chose 141 due to the size of them. And then we aligned. What we did is we took those blank chunks, those junction breakpoints, and then added an extra 50,000 base pairs on each side. So now we have a little portion of this synteny and this portion of this synteny and a break. And then we aligned all these with Mummer, a really good aligning program that we use a lot. And there are going to be 4 cases. We determined there'll be 4 cases what we see in the data. Of course, first, nothing aligned to this breakpoint. It's a draft genome. Let's see what we can find. Case 2, where a portion of the scaffold aligned to a portion of the junction. So it did map to a portion of this synteny, but then stopped in the middle of the breakpoint. Can't really tell anything from this data. We have something that goes over the entire junction, or something where there's actually 2. So we went through the data and we found some nice cases. This is case 4, where we have 2 scaffolds that cover the junction. This is one scaffold right here that covers, and you can see suddenly right here, it switches to a different scaffold. And then here's some nice case 3s where the entire scaffold covers the whole junction, and including the synteny. And I have this nice scaffold right here that goes over 2 whole places. Nice and long scaffold that we could see goes over both junction points and covers the entire 500 base pair for synteny. And we could see the same here. So overall, when we look at this, about 51% of the junctions that we looked at did have a case 3, which means the scaffold covered the entire junction point. So, and then the second most prominent was the 2 scaffolds. And of course, this could relate to the fact that it's a draft genome. We don't have enough sequencing to call. So this data suggests that the structural rearrangements happened early before speciation. Of course, this is a suggestion. We need a little bit more data, but it still it bodes confidence towards one direction. So next project. It is a very large international collaboration with many people, and including some public-private partnerships. I have to list here United Soybean Board, Bayer Crop Science, Dow Ag Science, and Monsanto. And it is the US Soybean Germplasm Sequencing Project through the University of Missouri. And through this project, their goal is to make a new reference assembly for Glycine Max, new reference genomes for Glycine Max cultivar called Lee, and a new soybean soja reference genomes, with the overarching goal of actually starting to doing a pan-genome data analysis with all these different genomes. So first of all, probably your first question is like, how in the world did we get to 4? Because Soybase has 2. So just to clear up confusion, we first had 1 that was released in 2010, and then 2 came out in 2013 and is available as Soybase and Phylosome. And then 3 was released by Hudson Alpha. And basically, it's the same structure as Glycine Max version 2, but it's just gap-filled. So clear up that confusion. Now the next one's going to be 4. So Glycine Max Lee and Glycine Soja. This will be the official community-supported assembly names. And we can see the Lee is a southern US cultivar. It is supposed to be a complement to the Glycine Max Williams 82. And then the G Soja was found to be the most genetically diverse in the Soy SNP 50K project. There already is graphical maps available for it, and it's well characterized. So it was a good candidate for a Glycine Soja reference map. And here's some numbers from the, again, these are still in draft stages, from the draft analysis. And so here's 3 for a reference point. The total number of base pairs that has been anchored into the molecules. And you can see there between Glycine Max Lee and Soja that they're actually a little bit larger than Glycine Max. That shows that there's more sequence. This bodes interesting. This could be interesting. So it's, of course, something that we look into using genome assembly structure comparisons. I hope you can see that in the back. So here's one. For example, on the bottom, I have G Soja. This is pseudomolecule 4. And here's Glycine Max version 3 against 4. And you can see it looks pretty normal. It looks like they match pretty well. But then things get interesting. We can see here, this is now chromosome 19 from G Soja. And this is from version 3, 19. We have an inversion here and a rearrangement between the 2 species. When we look at Lee and Glycine Max, we have a nice inversion. And then things get interesting again in 11, where we actually have an insertion. Actually, we have 2 insertions, one here, and then another rearrangement. So these are interesting structural concepts to look in the genome that bodes more looking at. So the Williams 82 is still being worked on. So we have these 3, and hopefully 4 will be much better than the rest, because the researchers are using optical mapping for this particular work. So the plan is basically it's similar to barcoding for when you're making a barcoding map when you're trying to organize all your different scaffolds. So just like similarly, use barcoding, use a die, and lay it all out to make this really nice consensus optical map. So this is from one of the collaborators in the project from the Chinese University of Hong Kong. and I will do my best to go through all this. So the red is the actual sequence that we have from the genome assembly. And these markers here, again, are basically the black lines are the barcode markers. And this is our optical map on the bottom. The blue is basically holes in between or missing data. So we can see here that these markers in a sequence show an inversion basically in the optical map. So the optical map was able to correct an inversion that was in the original scaffolding during the basically found a misassembly. It also found a breakpoint, what the collaborators are calling a breakpoint. But if you really look at it, there is no markers in the optical map here. This actually is another sign of a misassembly that can be fixed later. So using optical mapping will really help make version 4 a really nice resource for soybean researchers. So I bet you're all asking, okay, great, yeah, where is it? Where's the data? You're talking about this. Just give me the data so I can finish my research. Well, it's not done yet. I'm sorry. So the initial draft versions will be made publicly available before NCBI submission or before publication. But we ask that you follow the Fort Lauderdale Agreement and the estimated dates. Version 4, early 2018. That could mean anytime in 2018. You know how research goes. And then the Lee and the Soyja, hopefully soon. And when you're thinking hopefully soon is like, how am I going to find this out? So just to let you know and to pitch for Soybase. Soybase does have a number of ways that you can follow it up. Check the website. They have, for the graduate students, they had a Twitter feed, they got Facebook. Things like this will be posted available for you to find out. So, and with that, just an overview. The Australian wild relatives suggest structural rearrangements early, and that some wild species are evolving faster than others, and there will be future genome assemblies, and thank you for your time. Oh,
Jackie did great keeping the time. Just let you other speakers know. Does anybody have any questions?
Michelle?
As an Australian, I have an interest in these wild relatives. So did you find that there were particular genes that were being selected against when you were using your expression data and all of that stuff?
We don't really have enough to do expression data, but what we hope to look into, and I didn't have here, is basically looking at gene family analysis. Has there particular gene families that have expanded? Because since these are since it's a draft genome and it's transcriptome assemblies, we're limited on what we can call. So we know in previous studies that, yeah, there is some genes that have been expanded. Jeff Doyle and his group have done a lot of work with this, and they have proven that there are some that have expanded. So when we're doing the gene family analysis, we would expect to see some expanded. So that's what we hope to see, but we're still kind of going through that further analysis. So I will keep you updated on that one. Yes, ma'am?
Checking in, it's fascinating the difference in the evolution speed between some. Would you have some ideas or estimates or responding to what those wild species have evolved faster? Climatic variation, environmental stresses.
Let me go to that real quick to show. Where is it? So we know, like, for example, for these two, this one and this one, I can't really make that much of a call, but I know between the tomatella and the Dodgecarpa, this one was particularly interesting because Dodgecarpa is a tetraploid, first of all, which means, like, really? And then tomatella, the reason why it has a D3 next to it is because there's a tomatella, basically a complex of a tomatella species. Some of them are triploid, some of them are diploid. And this one was labeled D3 because it's diploid 3. What we did find, basically this whole structure is similar to what was found in previous studies. So that made us give clearances. But why exactly some are evolving faster, I cannot absolutely say. I can have suggestions and ideas. But, and then knowing where these grow, Tomatella is more tropical. As you saw, it went to Papua New Guinea, whereas the others, the other three, are not as tropical. They didn't move into Papua New Guinea, so it could be a niche thing, like basically an environmental niche. But this is, again, just basically poses more questions than answers.
And could I ask another question?
Yeah.
And could you elaborate on the time frame that those evolutionary changes are occurring?
I can't really elaborate. I don't have enough data to really say. I can make, basically we know from marks here that this is approximately 5 million years ago when the species split. I cannot remember off the top of my hand, and it's still been working on like how and when did these really start developing. I think it's something that it's still been worked on, and a case of just not enough data. Is like, again, it's a draft genome, they're only transcriptomes, and it's one of those cases where you only have so much money for your project.
And my next question. What about the synteny between those one species that are past evolutionary types with Glycine max?
I have not looked into that. That's a good thing to look into. This is all transcriptome data, so I can basically look at like genome synteny, like is there really major differences between the genes, but I cannot do like genome synteny because I don't have genomes for those.
Thank you, Brenda.
Thanks, Jackie, again.